Hi everyone, today is 2nd of March and this is our first lection of the course Neurophysiology and Cognitive Sciences. And today we'll have a kind of introduction, so I'll tell you about myself, some words about the organization of the course, a little overview of cognitive science, and we will remind some basics. So this is the plan for today. So my name is Ksenia Solovyova, and I have a PhD in computational neuroscience. On the one hand, I'm interested in understanding the mechanisms of the brain, and on the other hand, I'm interested in what are the states of the brain and how you can control the transition from one state to another state. Therefore, I work not only in academic science, but also I collaborate with some startups in the field of digital health and mental health. So you probably know but now there are a lot of publications in neurosciences and cognitive sciences, and there is a huge amount of empirical knowledge and data and about the brain. So you can find fMRI data, EEG data, and so on and so forth. So in my opinion, there is not, we haven't got enough good functional models of the brain. So we have a lot of empirical data, but not quite good brain models. So I mean such a model that we could build individually according to the brain of a specific person, for example, of my brain, of your brains. And yet this model would tell us something about the functioning of our brains. So when we have such model, we can understand the current state of the brain, of the consciousness, and how can we translate a person from one state to another state. And I would divide the states and modes of operation of the brain into normal, left row, or ordinary states, and altered ones. So I mean by ordinary states, those states in which each of us are often. So a high percentage of the population in their ordinary lives. So we can be productive or not productive. We can have different emotions, fear and happiness. We can be in depression, in an anxiety, or in a good mood in concentration. And altered states are not standard modes of operation of the brain. For example, some specific modes like meditation or mental disorders or lucid dreaming or hypnosis or so on and so forth. So what do you think is this division good or not very useful?
Well, what are we trying to reach? What is the target so we can know if this two paths are good or not? Like, what is this brain model for? What are these dots representing? So maybe you can have a fair judgment of this.
Okay, as I said, there is no good model of the brain, and maybe but I wish we will have such a model. So it's my division of my personal works. So these dots can be, for example, different states, different emotions. For example, maybe the green one some connected, for example, with concentration and with some specific areas of the brain or some specific nets of the brain. Maybe this model can represent the personality of a person. For example, I can be in this state or in this state or in this, and I can go from one state to another state, and some other person have his own transitions from one state to another state. Okay. And for altered states, we will have maybe some dots very far from the main core of the brain, because we can get these states very rarely. Okay.
Yeah. Okay.
In one of the projects, we have a research of productivity. The main idea of such studies is that your state of concentration or joy depends, among other things, on your biochemistry or the level of glucose, and then maybe on the enrichment of the brain with the oxygen. And in such kind of research, we divided all people in different groups. For example, a control group, sport group, meditation group, and maybe diet group. The participants have exercises or meditation during 1 or 2 months. It depends on the study. And before and after that, we measured their biochemistry, their cognitive functions with the help of some tests, and their patterns of EEG, so the patterns of their brain activity. As a result of, for example, as a result of this first study, we have found that in the very beginning, meditation or sport or cardio is better than a diet for cognitive functions, because after a diet, some cognitive functions are lower than after meditation and cardio, and as if the oxygen level of the prefrontal cortex. Another of our projects is Fuji Experience project, and it's related to the objectification of the process of meditation. So we have made a tool to visualize brain activity using real-time EEG, and counted some parameters to distinguish these altered states from normal sitting with eyes closed. You know, sometimes people sit and close their eyes, and they think that they meditate, but if we compare their state with just sitting and close their eyes, they are very similar. But the main problem with learning meditation is actually that all meditations are different. Even Buddhist loving kindness meditation and Vipassana meditation, they're different in terms of practice and in terms of, for example, EEG visualization. And now my main focus of work is related to the study of causal models of the brain and their application to mental disorders. So the main idea here is that correlation is not causation, and we can get data of healthy and ill brain, for example, fMRI data, and to create a graph model according to this causal causal structure, and we can find we can find if this causal structure of healthy brain are similar or are not similar as the brains of people, for example, with autism or Alzheimer's disease or so on. And the other thing is that measuring devices are slower than the processes in the brain. I mean, for example, for fMRI. Therefore, we can observe not true causal systems, but systems with kind of undersampling rate where there are more connections than in the initial system. Like here, on the first graph, we have got, this is a kind of model graph, and on the first picture, we have got real causal relation. On the second picture, we connect the nodes or the dots if we have got a path of the length 2. On the third picture, we have got a connection between nodes if we have got a path of the length 3 here, and so on. So, for example, if the real frequency of the parts of the brain is about 100 milliseconds, so every 100 of milliseconds, one part of the brain connects with another part of the brain, and we can measure this process only every 300 seconds. We can see this picture instead of the real one. And we will talk a little about this causal events, but this course was organized in 2020 with Nikolai Bushkov, and we tried to combine elements of neuroscience and cognitive science. And in the majority of lectures, I will talk about well-known facts from textbooks and about ideas from modern science, so from current articles. Okay, now are some words about the organization. This is our schedule. So we are going to have 8 lectures and one lecture for an exam. The white circles are online lectures, like now. The yellow circles will be organized in MIPT, maybe somewhere in the main building, and an exam also will be online, but I hope you will do all the tests and you won't need the exam, and I'll tell about this later. These are our topics. The first one is introduction, some organizational matters, some main things about neuroscience and cognitive neuroscience, and about brain, neurons, and so on. Then we'll have a lecture about basic neurophysiology, about neurons, synapses, about resting state potential and action potential. So it will be mostly about the neurons, so the lowest level. Then we will have a lecture about brain research methods, EEG, MEG, MRI, fMRI, and so on. Some history of these methods and the difference between them, how can we use them. Then we'll have a lecture about basic neuroanatomy. You know, there are a lot of different parts of the brain, and we know something about their functions and about their connections with each other. But I think if we want to use the information about these brain parts, we should have a separate lecture about them. Then we'll have a lecture about attention, sleep, and wakefulness. So about all these mechanisms connected with attention, some special brain networks, and sleep phases, and physiology of sleep. After that, we are going to have the lecture about brain networks. Then there we will have two parts. One part about brain networks in the meaning of parts of the brain, and another part will be about large-scale brain networks. Like default mode network, central executive network, salience network, and so on, which are very popular which are very popular in the last few years. Then we'll be closer to cognitive neuroscience. We'll have a lecture about thinking and imagination, about also about some mechanisms, and about insights, creativity, how can we find if a person is creative or not with the help of brain imaging. And the last one, the last lecture will be about some states of the mind, some ordinary states of the mind, and some altered states of the mind. Okay, and as you know, we have got a group in Telegram. I hope all of you are there, and you can ask any questions there in every time. And we will have the same link on our lectures in Zoom. And also, I'll send you this link for Google Classroom. There you will find the tests after every lecture. And also, I'll send the presentations and the recordings. And I think I also can download the recordings in YouTube. Do you know, what do you think? Is it convenient or not?
Yeah, it's fine.
Okay. Also, there are some textbooks. I've downloaded them in some special folder. I also will give you the link on this folder. So the main books are, for example, Gage and Barr's Fundamentals of Cognitive Neuroscience, Cognition, Brain, and Consciousness. Also, From Neuron to Brain, of Nichols and Kufler and so on. And for example, this book, Neuroscience, 5th edition, or this is a book of Dale Pearce, and the first two books are mostly about cognitive science, about thinking and these highest processes. And if you want to know more about the neurons, some details about neurophysiology and maybe the biochemistry and about neural channels, you should use these two books.
Doctor? Sorry, I want to ask, like, are we going to go into details on this course or just like general information about different topics?
You can find some additional information in these books, so they are not necessary.
But the information required from us, is it too much, or because this course like is not directly related to our biotechnology? So is it general information or details? What we have to study, I mean.
Okay, you can find almost all the answers on the tests in the lectures. But
okay,
if you want to know deeper, you can read these books. Just
okay.
Professor,
Thank
please, that means we will study from your presentations, or we need to go on and search for more information on this book.
yeah, so you can find all the necessary information in the presentation and in lectures, but if you don't understand some connections, maybe because we have got just 8 lectures, and so maybe some facts will be so you won't be able to understand the connections of some facts. And if you want to know deeper, you can read some articles in the books.
Okay, Professor, thank you.
Some supportive information. And some words about the grading process. I'll ask you to fill this form, just kind of registration form, some information about you, where you're from, and what do you want to know from this course, or what's your motivation of it. So just a few questions, maybe 3 of them, and you will have some quizzes after the lectures. They are not big, just 10 questions. And if you pass all these quizzes good enough, you won't have the final exam. Okay. So, yeah. But if in the case if your grade is not good enough for you, for example, after all these tests and quizzes, you have got I don't know 8, but do you think that you should have 9, for example? You can say, oh, can I pass an exam? And that's not really hard. You just have to present some articles, so some scientific article about the brain or about the neural system, so something connected with our lectures or about cognitive science, and maybe something which you are interested in. For example, maybe someone is interested in some pharmacy connected with Alzheimer's disease, and you can present this topic or something else.
Excuse me, are these quizzes for the previous lecture or the lecture being taught like at the same time? So now, like in lecture number 2, we'll have a quiz for lecture number 1 or lecture number 2 itself.
So after the lecture number 1, so after I'm not sure you will get the quiz after this lecture because this is an introductory lecture, but maybe you'll have a little test. But for example, after the lecture number 1, you will have a test, and you should do this test before the second lecture.
So we'll have it to do it like until the next week.
Yeah.
Okay.
But that's not so necessary. So if you haven't got time at all, you can pass it later. But that's not really good for your understanding the topics. So I hope you will do the tests before the next lecture.
And will we be doing any practical application for this? Like can we use any, because I've noticed that you opened Python, I guess. So I mean, like, will we be doing some codes related to this neuro and cognitive?
So you mean some practical lessons with EEG or something like that?
Yeah, because when you said there is offline classes, I noticed, or I just said like maybe it will be practical, or is it just like this?
If you want, it can be practical because I have some devices for measuring EEG, so we can.
Yeah, of course, it would be so nice.
So you can mention it in the comments in this forum if you like it, and maybe if several persons are interested in measuring their EEG and
yes,
doing some calculations with the rhythms and EEG connectivity, we can do that.
Is it the same devices that they use in movies, like for lie detection thing? To know if someone is lying?
Yeah, I know this series, so I can show you.
Very good to know.
Our devices. So wait a minute, maybe here. Wait a minute, maybe.
Doctor, is it possible to switch the course from tissue engineering to this one? Do you know about that?
I don't know. You can ask your administration.
Okay.
So we'll have this. Do you see the picture of EEG device on the screen? No?
Yeah.
Yes, it's showing. Yes,
Yes,
it's like a bracelet.
I like the one on the right.
On the left, Europlay. So we have got these devices, but in fact they're similar to Muse or something like that, like these EEG devices. But in Muse you have got about 3 channels, and here you have got more, 6, and reference channel. Okay, so now some overview of this, some overview of neuroscience. So in 2000, 2012, when I started to study the brain, there was a new wave of enthusiasm around the world for the brain and for the artificial intelligence. So there were started such big brain simulation project as the Blue Brain in Europe, Human Brain Project, the Brain in the US, and so on. And the Human Connectome project was launched in 2009. I have a picture on the right here from the Human Connectome project. And thanks to this project, at the moment we know which part of the brain are connected to which part. But so far, much remains unclear. For example, we don't know which neurons connect to which one. So, you know, neurons are much smaller than the brain regions. So it's harder to find out the connections of all neurons. In addition, we can have connections between brain regions, but we can if we know the connections, it doesn't mean that we know the protocol of how the information is transmitted from one part of the brain to another part of the brain. So the connections doesn't mean the everything. At the same time, thanks to advances in machine learning, we had some works, some articles like this. Jack Gallant and his team have learned how to restore video clips that person watched from fMRI images. So on the left side, you can see the presented clip, and on the right side, you can see the clip which is reconstructed from the brain activity, so from the fMRI, and it was in 2011, 10 years, 11 years ago. And then they trained algorithms to recognize screen pixels from brain voxels. Okay, so before that, they showed many different videos to, for example, one person and to another person, and they created these connections between the pixels, the activity on the video, and the voxels in the brain. As a result, we have such blurry pictures, but quite similar to the original videos. So it's kind of reconstruction of the thoughts in some way. But these results are very impressive, but here we are still working with the brain as a black box. So we don't know the mechanisms of the brain, we just can reconstruct the picture which was seen from the brain activity. To describe the mechanisms or principles of the brain, the different sciences have developed different approaches. For example, very often some terms from psychology, so we can find them in the neuroscience, because neuroscientists, they always ask the question, where is it in the brain? And psychologists they don't need to find some terms in the brain. Now artificial intelligence and neuroscience have converged a bit due to the success in the field of deep learning, in the field of artificial neural networks. So you know that deep learning architectures are inspired by the organization of the cerebral cortex, but still the task of artificial intelligence is an artificial mind, but not the artificial brain. So it's the same as making an airplane instead of a bird's wings. So the brain is like a bird. So the bird can fly. It's like the brain can think, but maybe it's not necessary to fly as a bird, just to fly. So other principles can be used in general in artificial intelligence. So neuroscience is about the neural system and about the brain. And in neuroscience, unlike psychology or philosophy, you cannot throw out the material substrate. Nevertheless, there are different branches of neuroscience. For example, behavioral neuroscience focuses on describing behavior in terms of how the neural system functions. By behavioral, I mean, for example, working or doing something, so some processes of behavior. Cognitive neuroscience study the processes of thinking, perception, and decision making in terms of also in terms of how the brain works. Computational neuroscience is the modeling of processes occurring in the brain and the processes occurring in neurons and in neural ensembles. Finally, neuroengineering is a quite broad field, so this includes both the creation of neural interfaces and brain-computer communication systems, and also it includes the creation of some specific neuromorphic chips that work according to the principles of neurons, of neural tissue. In theory, everything is much easier than in practice. And if we consider in a historical view the development of areas related to cognitive neurosciences, we can get the following picture. So there are some findings, some events. So here you can see the time, years, and some fields. IT, algorithms, philosophy, physics, biology, and physical methods. As you can see, until the 50s, the methods by which it was possible to study the brain, in addition to philosophy, they were methods of electrophysiology and optical microscopy. So you could just measure some electrical activity in the brain or on the surface of the head, and you could see the neurons themselves. But after the 15th, there were significant advances in several areas related to cognitive neuroscience. Here we have got the discovery of DNA, the discovery of principle for MRI, and then the principles of fMRI later, and the creation of first simple artificial neural network, which was called perceptron. And in fact, all these ideas and methods now produce a huge explosion of empirical data about the brain and about the principles of informational processing in neural systems. So we have got a lot of data on molecular biology of the brain and on different gene expressions. We have got a huge amount of data on fMRI, and you know all these things about artificial neural networks. And now some basics about the brain and about the neuroscience. So let's answer some questions. So what is the elementary particle of the brain? I think you should know that.
Neuron.
Neurons.
Yeah, of course, this is the neuron, and the neuron have a soma or a body of neuron, and also it have got axon and dendrites. And are there any other cells in the brain?
Yes, there is the supporting tissues.
There are some different cells, such as astrocytes, microglia, myelin shell, oligodendrocytes, and so on. And as you said, they have got some supportive functions, so they help neurons to get glucose and protect them, and also they help to produce the signal. Okay, so the axon can be covered by a myelin snell, but not every neuron have got the myelin. It's a kind of fatty substance. And so the other question is, what is the main difference between a neuron and a normal cell in the body?
Neurons
Nucleus.
doesn't have centrosomes. They don't divide.
I think in usual it has a nucleus and it can to differentiate like somatic.
Yeah, it's a good answer about the differentiation, but in fact, for example, a cell of a skin also can't differentiate. So if it's already differentiated. Okay, so there is some other differences, and neurons are excitable cells. By the way, there are some other excitable cells in the body. These are muscle cells, and so muscle cells are also excitable. And all cells in the body, they are connected with neighboring cells. But the neurons, they are not connected with all neighboring cells. So they have got just only connections in some specific places. And you know, we call them synapses. So the connections between neurons. The next fact is that there are neurotransmitters in the brain, and neurotransmitters are chemical messengers of the neural system, and different types of different types of neurons can release different types of neurotransmitters, and so they have different functions. There is an idea of biological perspective, and that's a view that all the mental disorders like depression and schizophrenia and so on are connected with this imbalance of different neurotransmitters. So there are many different types of neurotransmitters like dopamine, glutamate, serotonin, and so on. But do you know what is the main neurotransmitter in the brain? Kind of tricky question.
Acetylcholine.
Please,
No.
don't tell at the moment. One by one.
I think it's acetylcholine.
Yeah, definitely. I think so.
Because it's in the first line, yeah?
No,
because of the involving. Okay.
I don't know, but I heard this information before, but I don't know if it's correct or not.
We can't work here. All of them, of course, are necessary, but I usually call glutamate as the main neurotransmitter. You know why?
No.
Because 90%, so almost all of the neurons in the brain are glutamate neurons. Okay. So almost all of the excitatory neurons are glutamate. They produce the glutamate. And the GABA neurons are the main inhibitory. Yeah, the main inhibitory neurons. And you know, very often you can buy, for example, GABA in tablets. If you have an anxiety, so some people use it. Okay, and let's go down. So this is the synapse. You know, the synapse is the space between the terminal butter of one neuron and the dendrite of another neuron. And in this picture, you can see pseudo-colored image from a scanning electron microscope. Of course, the real molecules are not such colorful. Okay, so another necessary idea about the neurons and neural science is a resting potential. And we will discuss the resting potential on our next lecture in more details about the mechanisms of resting potential. But in fact, these blue pentagons, so natrium, is more highly concentrated outside the cell in extracellular fluid shown in blue, whereas potassium or purple squares, they are more highly concentrated near the membrane in the cytoplasm or in intracellular fluid. Other molecules, such as chloride ions, yellow circles, they are negatively charged, and negatively charged proteins, the brown squares, help contribute to a positive net charge in the extracellular fluid and a negative net charge in the intracellular fluid. Another important idea about the brain functioning and neural function is action potential. And we have got here some basic definitions. They are depolarization. So depolarization is membrane potential becomes less negative, making the neurons more likely to fire or excitation, and hyperpolarization. So membrane potential becomes more negative, making the neuron less likely to fire or inhibition. So with the help of GABA, we can have a hyperpolarization, and with the help of glutamate, we can have the depolarization. And the other two definitions are threshold of excitation. So the threshold of excitation is the level of charge in the membrane that causes the neuron to become active, and action potential, it's an electrical signal itself. During the action potential, the electrical charge across the membrane changes dramatically. So if the resting potential is minus 17 millivolts, and in the peak of action potential, we have got plus 30 millivolts. And this is the wave we can see in every point of axon. And so every neuron has an axon and the dendrites. The axon is usually the longest part of the neuron, and along the axon, the action potential is transmitted to another neurons. And the axon of the presynaptic neuron connects to the dendrite of the postsynaptic neuron, where the neurotransmitter is transmitted. So the presynaptic is a sending cell, and postsynaptic is a receiving cell. But the result of summation of postsynaptic potentials is the overall change in the membrane potential. So sometimes one signal from one presynaptic cell is not enough to excite the postsynaptic or receiving cell. That's why. So here you can see the green lines. The green
It's
line
not enough to excite it, the next synapse.
Here you have got green synapses of excitatory cells, or maybe glutamatic cells, and red synapses of maybe GABA cells from some inhibition cell. And in SOMA, or in axon hillock, we summarize all these EPSP and IPSPs. And only when we have got a threshold after the summation, we have got the action potential of our postsynaptic neuron. So to excite a postsynaptic neuron, a single signal from one presynaptic neuron is not always enough. So the excitation of a neuron is the result of the summation of influences from several neurons, or a result of summation of very frequent signals from one neuron, maybe. So once an action potential has occurred, excess neurotransmitters in the synapse either drift away, broken down, or are reabsorbed. So reuptake involves moving a neurotransmitter from the synapse back into the axon from which it was released. And reuptake involves moving neurotransmitters from the synapse back to axon. Okay, and some, you know that in neural system there are two main parts, so the central neural system and the peripheral.
Excuse me, Xenia, if I can stop you. I didn't get what when the uptake happens, like after the synapse happens, but the neurotransmitter does not go to the post-synaptic cell. It goes back to the first cell again, the first neuron. I didn't get what the uptake is about.
Okay, so after the neurotransmitter is released, the process of reuptake, so we always have the reuptake process, but some molecules can be held by the receptors of postsynaptic neuron. So it's kind of usual process. We usually use the same, almost the same molecules here.
So maybe it is too many for the docking site on the other cell, so they go back like this.
Yeah, they go back, but if we have got some special drugs, they can't go back. So there are some special drugs from reuptake, but in usual process they go back.
Okay.
Okay, so neural system is divided on central, it's brain and spinal cord, and peripheral neural system. And peripheral neural system is divided by somatic and autonomic or self-regulated neural system, which is divided on sympathetic and parasympathetic, or we also call them arousing and calming. And the brain also is divided on many different parts, such as cerebellum, brainstem, pons, hypothalamus, cerebral cortex, and in cerebral cortex we have got different lobes: frontal lobe, occipital lobe, temporal lobe, and parietal lobe. But we will discuss the anatomy of the brain later on our lecture about the brain anatomy. So for today, enough for the introductory lecture. See you after a week, and we will talk about some neurophysiology, about we will talk more about resting state mechanisms, about action potentials, and maybe about small neural networks. Okay, so see you after a week. Have a good neural connections. Goodbye.
Thank you.